Okay, welcome. My name is Krista Sipp, and I am a programs manager here at CanDoMS. I'm really excited to welcome you to Webinar Wednesday tonight. It is our final webinar of 2023. Tonight's topic is going to be 2023 Research Roundup. So we're going to go through all the research that has come about this year and keep you guys well informed as well as ourselves. So I will be your moderator this evening, and I do want to start off by thanking all of our sponsors. Without our sponsors, we wouldn't be able to do these programs. So thank you very much to all of our sponsors this evening. And without further ado, I'd love to introduce our speakers. So please welcome our speakers, Kathy Costello and Kalina Sanders. Welcome, ladies. Kathy's a nurse practitioner who's been working in MS since the 80s, and Kathy is also our Chief Operating Officer here at CanDoMS. Also joining us is Kalina Sanders, who's a neurologist specializing in MS. She's based in Jacksonville, Florida, and stays well informed on cutting-edge treatments and clinical research, so she is going to share a wealth of knowledge with us tonight. So ladies, I'm going to go ahead and hand the presentation off to you this evening. And I will take my leave. So thank you so much for joining us tonight and sharing all of your knowledge.
Thanks so much, Krista. And Kalina, it's so good to work with you again. I'm so happy that we have this opportunity. It's been great planning for this, and thanks for being here. I appreciate it. And thanks to everybody for joining us this evening. This is one of my favorite webinars, having been in academic medicine for many, many years. I always like to know what's new, what's coming down the pike, what do people have to look forward to in terms of not just treatments for MS in terms of medicines, but what other kinds of things are going on. And as you can see from our research roundup topics, we are going to talk about a class of drug that we've talked about before. That's the Bruton tyrosine kinase inhibitors, or the Bruton or the BKTIs. And we will talk about biosimilars and touch a little bit on generics. We'll talk about a new drug that's actually completed phase 1. So it's very early, Foralumab. We're going to talk a little bit about lifestyle, which again, exercise and diet. There are some new papers that were published this year that give some even added strength to what we know about activity and diet. And we're going to talk about biomarkers. There's been a tremendous amount of research into biomarkers. How can we better monitor what's happening in MS? And we're even going to talk a little bit about the use of AI and MRIs, which is very interesting. I know there's going to be a lot of questions. Krista is going to be monitoring the chat on our behalf and sending me messages about what questions have come up. We're going to do our darndest to get to all the questions, but it's unlikely that we will, despite our best efforts. Having said that, we really do encourage you, and you can do it tonight. Get on your email. Chris is going to drop the email into the chat, and you can email your questions. While I might not get to them tonight, here's how it's going to go. They're going to come to me, and I'm going to send them to Kalina, and then I'm going to get those answers back, and out they're going to go to everybody. So or to the person who has written in the question. So we'll do our best, but that is a mechanism if you have a burning question, and we will definitely do our best. I see a question already that we're asking if there's any handout. We will have a handout. It's going to be dropped in the chat. It's going to be specific to MRI, because we feel that's the one thing that is a guideline that you can actually take to your provider and share, that gives some idea of what kind of MRIs are recommended, when they're anticipated to be done or recommended to be done. So you will receive that, and that will come up in the chat when we get to that part of our conversation. So I think I've covered everything we need to in our housekeeping, and I think we ought to just get right to it because there's a lot on our agenda. And we are going to start with BTKIs. And there's a lot of interesting news about BTKI, some of which actually happened today. But let's talk first, Kalina, if okay with you. We've heard a lot about how these drugs actually work inside the central nervous system. These are oral medications. There's a lot of different ones under investigation as we speak. but they're working inside the central nervous system. Can you talk a little bit about why that might be important and maybe touch on that topic that we're hearing a lot about called smoldering MS?
Thank you, Kathy. And I'd like to take a moment to say I'm so excited to be here tonight to share some valuable information. Kathy and I have been friends for a very long time, so it's always good to share the platform with you. And you're right, we have a lot of exciting things, starting with smoldering MS, which is kind of a new buzz term, but really explaining an old thought that we've had about multiple sclerosis. So when we think about multiple sclerosis, we think that there are compartmentalized or acute inflammatory attacks. And historically, we felt that there was a secondary neurodegenerative process. And as pathology evaluations and testing that has been done with more advanced MRIs, we're actually finding that there is a smoldering small amount of chronic inflammatory activity that is occurring throughout the central nervous system that is not associated with relapses. And when we think about the immune system and its impact on the nervous system, we know that our T cells and our B cells that are out in the periphery outside of the central nervous systems are big drivers for inflammatory disease. But what we haven't really explained a lot until here recently is that there are somewhat immune cells inside of the central nervous system. And those innate immune cells are called microglia. And when microglia are there and functioning well, they're really there to help to support the cells that make the myelin and make sure that the environment is somewhat happy. And so when someone has more progressive disease, or they're having a term what we call PIRA, progression independent of relapse activity, we think that there is a dysfunction in the microglia inside of the central nervous system. And so that is where the BTKI, or the Bruton and tyrosine kinase inhibitors, have their impact. We know that the B cells and these microglia inside of the central nervous system has this enzyme called BTK. And the newer class of medications that are still under development are targeting that enzyme. And when they block or inhibit that enzyme, then B cells out in the periphery aren't able to proliferate or grow. And the microglia that are dysfunctional are also not able to grow or proliferate. And so because of that, we hope to see that the impact on progressive disease or smoldering MS will have a significant benefit with the use of BTKIs.
So if I have this right, then the smoldering process is not suddenly a new process that we've learned is going on. This is something that has been going on, but now we have a better understanding of why it's happening, which provides new targets for treatment. Do I have it?
Correct. You have it perfect.
Okay. So like everything else in MS, then the smoldering process is going to vary from person to person, correct? I mean, just like we know when we talk about progression, we see people who have more progression, some people have less progression. So the smoldering process being really what's going on to cause this progression will vary amongst people.
Correct.
Okay.
MS is still the snowflake disease, right? There are no two MS patients alike. And you are right. Some people will have varying degrees of smoldering activity within their central nervous system. They will have varying degrees of reserve, which is also important for function over time. So there are a number of different factors in addition to microglial activation and the smoldering MS that actually result in some of the symptoms that we see, such as fatigue, cognitive dysfunction, and obviously the mobility decline.
Yeah. So for people, what I remembered from clinical practice is people would have an MRI done. Say they'd had MS for, I don't know, 8, 10, 12 years. They'd have an MRI done. The MRI looked exactly the same. And we'd be so excited. Oh, look, isn't this wonderful? Your MRI looks great. Isn't that what? And they think, well, yeah, thanks, but I don't feel so great. I'm feeling worse. And so this is what's going behind that, what we see as a normal MRI, but this is what's happening within the central nervous system to cause that kind of progression.
Correct. And so the current MRI magnet strengths that we commonly used in clinical practice are probably not strong enough to really see and pick up on this smoldering MS. The other thing is many of the radiologists that are reading these scans are not familiar with it and are not only lookout for it, but we know it exists. There are some higher magnet MRIs that are utilized in research centers and other academic facilities that are helping us to hone in on it. And I think that's also what's going to help clinical trial completion, and particularly with the BTK inhibitors, which are small enough to actually enter the central nervous system, which is different than our other disease modifying therapies, where it's felt that there is very little impact inside of the central nervous system.
Yeah, interesting. So back to the BTKIs. These are oral drugs. They're small molecules. They're in phase 3, mostly. Some are still, I guess, in phase 2, 3 research. And for everyone who probably knows this, but for phase 3 research, that's where it's several hundred people, up to thousands of people given some drugs. Sometimes 2 different trials need to be done in phase 3 before there's sufficient data for the FDA to evaluate. So a company does a trial, they analyze their data, and they decide if they want to submit to the FDA for potential approval. And then the FDA goes through all of the data to see if it really holds up. Does it hold up in terms of efficacy? Does it hold up in terms of safety? And so what's happening right now with the BTKIs? Where are they?
So there are 5 BTK inhibitors that are under investigation currently, and I'm going to try my best not to butcher them, but they're evobrutinib, tolebrutinib, fenebrutinib, orelabrutinib, and remibrutinib. I did do it. And they're all, as Kathy just mentioned, in different phases of their clinical evaluations or assessments. Some of them are completing their phase 3 clinical trials. And as Kathy alluded to earlier on in the introduction, we had some late breaking news actually here today regarding evobrutinib, in which in their 2 clinical trials that were looking to treat relapsing forms of multiple sclerosis, where it was compared to teriflunomide or Aubagio. It did not meet its clinical endpoints with reducing annualized relapse rates. But what that truly means is that they were not able to show any difference between themselves and teriflunomide. Both drugs were effective, but their goal was to determine whether or not they had superior efficacy to teriflunomide, and they had very similar, fairly low annualized relapse rates. The fenebrutinib also had some recent late-breaking news as well, in which the FDA has placed a hold temporarily on clinical trial continuation due to 2 cases of elevated liver enzymes. The other 3 are still ongoing in their clinical trials, where they're assessing efficacy or effectiveness in not just relapsing MS, but also primary progressive MS and non-active secondary progressive MS. And so as we were talking earlier about the microglia and their impact on smoldering MS, the real thought is that this class of medication may have its best effectiveness against progressive disease and maybe not so much the relapsing MS.
Yeah. So there's been a couple questions, Kalina, just about how the drug works, its mechanism of action. We've talked about reducing activation of microglia, which can contribute to that smoldering process. We've talked about blocking the maturation of B cells, which are also believed to be operating inside the central nervous system. Do these have any effect on remyelination? Is there any hope there? Or is it really just to stop the process or to slow the process that's happening?
Correct. They are not mechanistically able to promote remyelination, which is done by a different cell within our central nervous system.
Right. So that they're not going to stop, I should rephrase that. They're not going to repair the damage, but they can slow down that process. And, you know, potentially they can maybe stop it. We don't have that information as yet, but at the very least, we would anticipate a slowing down of that progressive process that's being driven by this smoldering MS. Okay, very good. All right. So if one was to be approved, we're still a little ways off, right? We're probably at least another year away, would you say?
Correct. The late breaking news that we had today is really early data analysis. The Merck, the company that makes the ibrutinib, they noted that they still have to do their final analysis. So this process definitely does take time. And generally, when a application is placed in front of the FDA for approval, that takes another 6 to 10 months before they provide an answer. So we are at the earliest of 2025.
Okay. All right. Well, there's a message here. Will another 100 years go by? I certainly hope not. I don't have another 100 years in this profession, and people on this webinar don't have another 100 years. So I think that as fast as the research can go and still make sure that whatever drug comes out is truly not only effective, but also safe. The good news with this is knowing that there's additional targets that will drive additional research into different types of treatments. And I think that is true. And researchers are looking at that, because what researchers are now interested in is not so much that inflammatory process. As it turns out, we've got pretty darn good drugs for inflammation. The unmet need is, what do we do about this progressive process that's still happening? And so I think a lot of researchers have turned their attention there, and that's a good thing as far as I'm concerned. There's another drug that's actually at the FDA for evaluation, and it's actually kind of an old drug, but with a new twist. And that's an extended release Glatiramer acetate. And Glatiramer acetate is Copaxone, or was Copaxone. Now there's lots of different formulations, generics of Copaxone. There's Glatopa, Glatiramer acetate for injection, and now GA Depot, which, Kalina, tell us a little bit about this.
Sure. So for all of my super responders to Copaxone that are maybe getting needle fatigue or wanting to switch to an agent that has a better safety profile, particularly if they're risk averse, GA Depot completed its phase 3 clinical trial, where it had a significant reduction in annualized relapse rates. It is an intramuscular injection, but it's given once a month, as opposed to 3 times a week or even every day. It, in the clinical trials, had the same similar adverse event profile as we know and are familiar with Glatiramer acetate or Copaxone. The FDA is supposed to provide an answer to the company in March of next year.
Okay, so coming up, that's only a few months away. Obviously, it's not going to be for everybody, but there are going to be people who are on GA now, but maybe people who are contemplating getting pregnant. It's a very safe drug. It doesn't have a lot of side effects or risks, so there will be a place for this in our armamentarium, which I think is fantastic. And once a month beats the heck out of what that used to be, which was every day, right? So anyway, all right, let's move on because we're going to get front loaded as we usually do. GA is, by the way, for relapsing forms of MS, right?
Correct.
So let's talk a little bit about biosimilars. There was a big approval in August of a drug called Tyruko, which is a biosimilar to natalizumab. The trade name is Tysabre. It's FDA approved and it will be available early in 2024. Can you walk us through the FDA approval process for a biosimilar? Because I think that there's always concern, is this going to be good enough, as good, side effects the same, those kinds of things. Can you run us through that?
Definitely. The biosimilars are only available or occur or develop, with biologics. So I think it's important for us to understand the distinction between how our drugs are made. Many of our drugs are made chemically in a laboratory, and they're able to be reproduced very easily. Medications like natalizumab are actually biologics in which they use live organisms to grow and create the medication. So the process for biosimilars is much different than what we would see with generic approvals. So for a biosimilar to be approved, they have to first characterize their reference product and really understand its variability and the identified targets. And then they have to develop their own live organism cell line and manufacturing process. They're unable to take the cell line from the reference product, but they must create their own. And after they do that, then they should establish similarity. And different than generics, this actually requires clinical trial data. So they have to show that their cell line and product is not only as effective, but as safe as the reference product. So when we look through and look at how the drug companies are looking at biologics, really the bulk of their process is to understand the reference product, know how to grow their cell lines to create the product, and develop the manufacturing process. And then they throw a clinical trial on top of that.
Wow, that's a complicated process, but important to know. And from what I heard you say, there's variability in the reference product, meaning a biologic drug is a little different from quote unquote batch to batch, but it's similar enough. And I don't mean that in a negative sense, but the similarity is such that it doesn't change how the drug works. It doesn't change the side effects, doesn't change that. And the same must be true for the biosimilar, right? There will be variability. That's known. It's known in the reference product. So, but the variability is small enough that it doesn't change how the drug actually works and the anticipated effect of the drug. Is that correct?
That is definitely correct, Kathy. I think when you don't have the exact copy of the reference product from batch to batch, it's very difficult to have the exact product for biosimilars. But they definitely should show that they have similar efficacy and safety, which Tyruko, which was FDA approved in August, did show in a phase 3 clinical trial compared to natalizumab. And after 24 and 48 weeks, they had very similar reduction in relapse rates. Their disability progression was minimal in both groups, but very similar. And then they also found that the adverse events were similar compared to natalizumab.
And that includes the risk for PML, correct?
Correct. That includes the risk for PML.
So PML, progressive multifocal leukoencephalopathy, is the infection of the brain that can occur in people who are on Tysabre. Other products too, but in this case, we're talking about natalizumab products. The same risks exist for Tyruko, and the company will have a test to monitor that. And so everyone's aware that that's not going away with this drug because it's a biosimilar, but the same attention will be paid by providers and also by the company to ensure that testing is available to pick it up if it would occur in someone. So that's important to know. I'm going to move on. And just one last thing. I just want people to know that while the process is different for generics and biosimilars, it doesn't mean that generics are lesser products. That's not the point. It's just different. When you have chemicals, you can use a recipe and you can put those chemicals together by the recipe and make a generic drug. That has to be exact. I mean, it has to be an exact replica of the chemical structure of the reference product. And it has to have the same way that it gets into the system, is metabolized in the system and excreted by the system. So that it's not to say that it's less than, it's just a different process, because we have quite a few generics now in the MS space. A lot of our oral medications are coming along with generics. And for any of us who are getting any product, antibiotics, hypertension drugs, any drugs, there's loads of generics that are out there.
So Kathy, I'll jump in and say biosimilars is not a new technology either. The FDA has approved, I think, 44 other biosimilars in other disease processes. So this is not unique to multiple sclerosis, and the FDA is not unfamiliar with how to process this type of approval. So that should also provide some reassurance as well.
Great point. So there's been a couple questions about progressive MS, primary progressive or secondary progressive MS. And interestingly enough, and maybe we didn't make it clear, one of the types of MS or the disease courses that the BTKIs are being thought to be useful for is progressive MS. And so their ability to work inside the brain may be the driver of their ability to impact that type of pathology. Right. So think of them as potentially for relapsing, but also potentially for progressive. And that brings me to the next item on our agenda, which is an anti-CD3 drug called Foralumab. So a monoclonal antibody, and it's early in its development, but it's given, are you ready for this one? Intranasally. So it's a nasal spray. That's amazing to me. Anyway. Take it away, Kalina. Tell us a little bit about Foralumab.
Yes. So as Kathy mentioned, it is a monoclonal antibody that targets T cells. So we've talked a lot about B cells tonight, but this is one that targets T cells that have a protein called CD3 on them. And when given to MS animal models, the nasal anti-CD3s tends to suppress disease by inducing regulatory T cells. So we have T cells that are there to help us out and regulate things. So blocking CD3 T cells, we are able to upregulate our regulatory T cells. And then they are also there and it also helps to dampen or reduce microglial inflammation. And so here again, we're hearing that key word of microglial, which again plays a significant role in smoldering MS and disability progression. It was studied in a kind of a point of concept trial in just 6 patients, and they were given the medication intranasally 3 times a week. They would do that for, I believe, 2 weeks, and then they'll take a week off, and then they'll do it 3 times a week, and then it was alternating. The first 2 patients that were enrolled in this trial have now been on it for more than a year. The other ones have been followed at least 6 months. So we, again, have some early data. One interesting thing about this trial, and not something that is commonly used in our other disease-modifying therapies, is they use PET scans to look to see whether or not there was microglial activity. And they found in these PET scans that after 6 months of treatment, 5 out of 6 patients showed a reduction in that microglial activity. So that was a marker to help to show us that this drug is doing exactly what we were expecting it to do. One patient did have an increase in their microglial activation, and that also still needs to be sorted out. In that clinical trial, I'm sorry, go ahead, Kathy.
No, it's just, it was interesting that one person had activation, but fortunately, most people did not. Interesting.
Correct. They also looked at EDSS, so that's one of our markers for disability progression in our clinical trials. And one patient in the clinical trial, I'm sorry, 2 patients in the clinical trial had a reduction in their disability status. And so they had potentially improvement in their function. And the remainder 4 people had stable EDSSs over the course of their treatment. So there's some clinical markers that this therapeutic target may help to slow down progression. They did note that it was well tolerated and no severe side effects, but this again was an early study, and they're looking to now plan a phase 2A clinical trial to continue the investigation in non-active secondary progressive MS patients.
So as I mentioned, and to the questions that have been asked, there's a lot of research going on looking at different targets, but also different drugs for progressive MS. And again, not the part of progressive MS that's still inflammatory. We're looking at non-inflammatory. So again, really looking at that underlying process that's going on inside the central nervous system. Foralumab is interesting. If I'm not mistaken, it's also being tested for other indications that aren't MS. Is that correct? What are some of those things?
You know, Kathy, I don't quite remember offhand. Yeah. I flew away.
I think I recall that it might have been diabetes was one of them, which really type 1 diabetes, and it might have been Alzheimer's. But I know that I guess what we have to think about is when researchers see targets that are similar in different diseases, they may see if these drugs are effective in these other diseases as well. For example, natalizumab or Tysabre has an indication in Crohn's disease. And so I think that when there's some similarity in the pathology or the immunopathology, then sometimes these drugs are tried for other indications. So that's a little ways off, but just an important point to know that researchers really are looking at progressive MS. And just one quick question before we get to diet and exercise. There's been a number of people, and to be perfectly honest, I'm interested in this too, that what happens when you get older? Okay, so now what happens when you're in your 60s or your 70s and your MS is still just kind of perking along? And what do? Are people okay to take all of these different drugs that work on the immune system? Are there cautions that we need to have when looking at an older person with MS?
When there are a number of issues that we have to consider as a person ages, right? We know that as a person gets older, their immune system is less likely to be inflammatory or active. They have what we call immunosenescence. And so we have to think about the mechanism of their MS at that point. Although we've been talking a lot about smoldering MS, and we know that our current disease-modifying therapies are very effective at reducing that inflammatory activity, maybe not as effective at dealing with that smoldering MS or the progressive nature of the disease. And so at that point, we do have to have that risk-benefit discussion. We have to weigh the potential benefits with the potential risk. And as a person gets older, they're at increased risk for primarily infection and other adverse reactions, such as liver abnormalities and other laboratory abnormalities. You think about it, having MS unfortunately doesn't prevent a patient from having other diseases. So they have comorbidities that require other medications. And so we have to consider all of these factors as a person gets older and have an individual discussion on the risk benefit ratio.
Yeah, it's concerning for people. I mean, I think that, I mean, seriously, when I think about it as a person getting older is not for sissies. And if you have MS and you're getting older, it's even more complicated. But I think what we turn to right now, as the immune system changes, there are things that we can take control of in our own lives that can be really beneficial. This is not like, oh, well, you can't do this, so do this. It's not like that. These are real interventions that we really need to optimize. So I'm going to shift to diet. And I don't like that word because it's kind of like a 4-letter word, actually. And you think of, oh, I got to be on a diet. I got to lose some weight. Oh, I got to be on a diet. My doctor told me I should lose some weight. That's not what I'm talking about here. I'm talking about thinking about nutrition and thinking about your lifestyle and how do you incorporate good nutrition into your life. And with that, I'd like you to talk to us about, we're still going to look at what kind of diet would be best for people, and is there recent research to support it? So I'm going to turn things over to you again, Kalina, on that.
Okay. I agree, Kathy. That word triggered a lot of emotions in me as well. Especially, as we all struggle to lose weight, we don't need another diet. We don't need another diet. But I like the terms, as you mentioned, lifestyle modification, eating habits, that really kind of explain the role of diet in multiple sclerosis. We know that healthy eating habits is important for our basic bodily function. Our bodies need electrolytes, it needs glucose and sugars, it needs vitamins derived from the foods that we eat. We also know that they have an impact on the functioning of the immune system. So there are certain foods, eating habits, or diets that are known to minimize the inflammatory process of our immune system. Although the evidence is mixed, it's generally recommended that persons living with MS really should adhere to an anti-inflammatory type of diet.
So what would that look like? Like the research that was done at Mount Sinai by Alana Katz-Sands presented this year, was looking at the Mediterranean diet. Would you consider that to be an anti-inflammatory diet? And what is her research showing in terms of the impact for MS?
The Mediterranean diet is, from a food perspective, is really a plant-based diet. It is high in vegetables, fruits, beans, nuts, and seeds, and they limit their amount of meat to primarily fish and chicken. But as you mentioned also, Kathy, earlier, it's a lifestyle. So part of this Mediterranean diet is to share meals with your family. And they also incorporate physical activity in this Mediterranean type of diet. And so what they studied in this clinical trial was they looked at those patients who self-reported whether or not they adhere to a Mediterranean diet. And they also measured their objective disability. And they found that those who adhered to the diet had better functional outcomes and physically as well as patient-reported outcomes. So they had decrease in disability status, fatigue. They felt that their walking was better, as well as mood disorders.
Wow. And am I correct? I think that there was a study done in a non-MS, just a general population. And not only was it was effective for preventing cardiovascular disease, there was better cognitive function. Am I right on that?
Yes, there was a recent review article that looked at the evidence of Mediterranean diet and its impact on cognitive decline. And it showed that there was a lower risk of developing Alzheimer's disease.
Wow, that's impressive. So again, for those of us that are getting older, these are important things. I mean, they're important because, as you mentioned, as we get older, if you have MS, you're also, you know what, it should, darn it, but it doesn't protect you from other things. So we need to attend to our entire lifestyle. So I appreciate that very much. So let's turn for a minute to exercise. It's not 4 letters, but it might as well be, because when we hear exercise, we hear, okay, go to the gym, sweat, feel like heck, but hey, it's good for you. But really what we're talking about, again, is incorporating activity into your life, right? Exercise is more structured, right? You're going to do a certain amount of things for a certain amount of time versus your normal activity level can vary from low activity to more activity. And we can get almost as much out of our activity as we can out of formal structured exercise. Having said that, there was an exercise study done that I'm going to ask you about in a second that was done around aerobic exercise. And it was done at the Cleveland Clinic, and they have a huge MS center there. And it was a really interesting study, and I'm going to let Dr. Sanders tell you about it because it's kind of cool.
It was a cool study. It was a pilot study that looked to determine whether or not aerobic cycling intervention had an impact on patients' symptoms and quality of life. And so they enrolled 18 patients in their study. There were 2 groups. There was one group that had a forced exercise rate of their bicycle. So their bicycle propelled them, and there was a motor behind it. And then there was another group that had a voluntary exercise where they were able to propel themselves. But both groups completed a 12-week moderate to high intensity aerobic cycling intervention. They had, I think, a session of about 45 minutes, and this was a stationary bike, so they weren't biking along, the roads. But both groups had 24 total sessions over that 12-week period, and their target aerobic intensity was around 60 to 80%. So they were biking during that study, and so they found that in both groups they had improvement in their physical fatigue, their overall mental fatigue, and they also found that their pain intensity and how it interfered in their life also decreased. And a small number of patients had improvement in their walking speed at the end of this 12-week session.
That's fantastic. So those that had the motor support, what my understanding is that even people who had mobility compromise were able to participate and increase their aerobic activity, particularly if they had that extra forced support. So it might open doors for people who otherwise would think, I can't do that. I just don't have that kind of mobility. So maybe not ready today, but it looks as though having that aerobic activity on a regular basis really could improve a lot of symptoms that people have. Pain is pretty darn prevalent. Probably a good 65 or 70% of people with MS have pain, and that would be fantastic to improve that. So the bottom line here is, and I'm going to say for diet, in the words of Michael Pollan, eat less, eat real food, eat more plants. So there you go. So that's not a diet, that's a lifestyle. And I think that covers the Mediterranean diet, right? If it comes in a bag, a box, or a can, be suspicious. It's processed. And that's where a lot of our inflammatory foods can be found. And for activity, I think the message is to be active, to what you're able to do. And it's going to vary based on what's going on with your MS and other things. But if you are able to be active, there are benefits that you will get from that activity. So lastly, and then hopefully if we can take about 5 minutes, we'll have 10 minutes to do nothing but questions. Let's talk about biomarkers. And there's been a huge amount of research over the last probably, I would say 6 to 8 to 10 years about biomarkers. What does this mean? How do we measure what's happening in MS? So we can certainly do a neurological exam, but are there other types of tests that we can do that can give us information about what's going on with your MS? Are you at risk to progress? Are you having inflammatory changes? Is your MS worsening based on these biomarkers? Because sometimes it would be nice to know before you're feeling worse. It would be nice to know if we have an idea that you're getting worse, because that could, of course, drive a treatment decision. So let's talk with the one that we probably don't think about anymore as being an exotic marker, but let's talk about MRI. That's an important biomarker, right?
It is, but MRIs has been and continues to be a very, very valuable biomarker for not only the diagnosis of MS, but the monitoring of the disease. It is highly correlated within the inflammatory aspect of MS, and it gives us information even when a person is clinically silent. So MRIs are extremely important. There have been some recent guidelines and recommendations on when and how a person should have an MRI. I think it's of particular importance in those who are needing follow-up MRIs. And thank you, they posted here. What are the key takeaways from our most recent guidelines? So everyone should obviously have an MRI at the time of their diagnosis, but once they're diagnosed, a baseline MRI should be done prior to initiating or switching to a new disease-modifying therapy. A re-baseline should also be done between 3 and 6 months after starting that new treatment. And then for those patients that are clinically stable, a surveillance scan should be done every 6 months. If you're really stable, we can go out to 2 years. Patients also, if you are pregnant and you've delivered, you should be re-baselined as well after your pregnancy. Now that's for patients who have relapsing disease. For patients with progressive MS, MRIs are done at diagnosis, but according to the recent guidelines, annual brain MRIs are not recommended. However, they should be done to reevaluate, particularly if there's an unexpected decline in your function or if you have new symptoms. That's when MRI should be performed if you have progressive disease. I would also like to add that they make recommendations for when gadolinium is used. Obviously, it's important for the diagnosis, but they also recommend that gadolinium be used on surveillance scans in the first year, particularly if you did not have a re-baseline at 3 or 6 months. They also recommend gadolinium if you have clinical activity or if you're switching to a different disease-modifying therapies. So you might find that your provider if you have a prior MRI to compare to, they may not have gadolinium on every surveillance MRI.
Gotcha. Thank you for that. And I think Krista just dropped the link to the Consortium of MS Centers updated MRI guidelines. So this looks like a very complicated document, but there are some scenarios that they describe in the document that illustrate exactly what Kalina was just going over. When do you get an MRI and what kinds of situations are MRIs appropriate? And, I think providers don't all know about these guidelines. You would think that that's possible. That's not possible, but it is. So I think it's fine to share these guidelines with your provider. Your provider can also share them with a radiologist because there's guidance for radiology and how MRIs are done. And so I think that these are very important. And as a new twist, we hear an awful lot about AI. Well, tell us a little bit about AI and MRIs for MS.
Yes, the infamous AI. I don't know whether to be excited or scared. I'm still trying to decide. But yeah, I know. We've had software to help us with utilizing and analyzing MRIs, but there is a new program that the FDA recently approved called Pixel.Neuro, and they actually have additional carve out for MS in their software profile. And so this is a software that uses AI to detect changes. It provides an analysis and a report. And guess what? less than 5 minutes, right? In line with what we see with AI, very quick responses. Obviously, I think that there will be further studies and validation forthcoming. And so I would like to add that the future is now here.
Oh, that's pretty incredible. And I think if we utilize that in conjunction with a knowledgeable radiologist opinion, I think that we're going to be a lot more precise with what we learn and glean from an MRI. But MRI isn't our only biomarker. We also have a few blood tests that are out there. One, we're going to talk about three. We're going to talk about NFL, not the National Football League, but we're going to talk about GFAP, and we're going to talk about the Octave Bioscience blood test. So let's start with serum NFL.
Yeah, so not the National Football League, but neurofilament light is what that stands for. And neurofilament is what they call a cytoskeletal protein that is specific to neurons. And this is very similar to the bones we have in our body. It's the protein that makes up the structure or the bones of an axon. And neurofilament light chain is the most abundant subunit, there's 4 of them, of neurofilaments. And so when there is destruction or damage to the axon, as if you were crushed, your bones will break apart, right? And they will travel. So does this neurofilament light chain. So it is released into the cerebral spinal fluid following axonal injury. And so it can also help us to capture the extent of how much damage that there is. It's obviously found in the spinal fluid, but we can also detect it in the blood. And so neurofilament light chain, although it's not specific to multiple sclerosis, it can be found in other diseases like stroke, Alzheimer's, Parkinson's. It is known to be elevated in patients with MS compared to non-MS patients. And the literature also suggests that progressive MS patients also have higher neurofilament lights compared to relapsing MS. And so further studies suggest that it is also elevated in relapses. Now, this is a test that is now available and can be ordered through any laboratory, Quest or LabCorp. And many providers are using it to help monitor disease activity over time and response to treatment. It's not a perfect test, but it's definitely growing in potential.
Interesting. So again, it won't be the test, but it will be part of the putting the puzzle together, and it will add important information to when we're trying to figure out if someone's at risk for progression. Do we need to consider making a treatment change? Those kinds of things could be very helpful. It won't be the only answer. Now, what about GFAP, or glial fibrillary acid protein? What about this one? And how does it fit in? Or alphabet soup.
Exactly. It is a protein that's found, again, in the cytoskeleton, but of astrocytes. And we've not talked about astrocytes, but astrocytes are cells within the central nervous system that provide support as well as protection, so that neuroprotection to our neurons. And damage to the astrocytes release GFAP into the spinal fluid and eventually into the blood. A recent study showed that GFAP levels were highest among worsening primary or progressive MS patients. And that's followed by active relapsing remitting MS. Elevated GFAP is associated with actually worsening of gray matter volume, so brain shrinkage, as well as future risk of progression independent of relapses. So we can think about GFAP as a useful biomarker for disease progression. So NFL, we think about inflammatory activity, acute relapses. And GFAP, we can think about maybe in the future helping us to assess progressive disease.
A welcome test for sure. So we'll be looking for that one. Finally, let's talk about that Octave Bioscience test. You're actually using this in your practice. So tell us a little bit about that, and then we'll see if we can get to at least a few questions because we've tried to cover a lot of ground.
Perfect. I'll be real quick because Octave is a test that looks at MS disease activity, and it is a clinically validated blood biomarker assay that includes 18 different biomarkers, but also neurofilament light and GFAP. And so based on the profile of the patient's biomarkers, they're able to provide them with a score, and it's divided into low, moderate, and high disease activity. And the score actually correlates strongly with the presence of GAD-enhancing lesions or new or enlarging T2 lesions. And so it is felt to be predictive when combined with MRI and clinical activity for future relapses. I think, there's a lot more that we need to learn and utilize it. I think it's very helpful. Again, I'm using it as a composite, and it's not the sole thing that I use to determine changes in disease modifying therapies, but I definitely have started to utilize it in combination with the other features of a patient.
Excellent. So it's still a puzzle that we have to put together, but some of these tools are coming along to help us maybe make quicker decisions, but also make more accurate assessments. And that is always going to be useful in our treatment decision making. So I'm happy that these things have come along. Now, I know that we probably have missed a bunch of questions. Krista, if we've missed anything really important, please feel free to jump on and let us know. I know we had a lot of questions about progressive MS, and I hope that we've covered that there's a lot of research out there. We touched on a few things that are there. We talked about what happens with aging. Oh, one question that came along with MRI was around gadolinium. I think that you actually answered that, but it's definitely used at the time of diagnosis, and it doesn't always need to be used as we're monitoring going forward. But it has value in letting us know about new inflammation, right? Gadolinium enhancement lasts for a short period of time, but it can indicate new inflammation. So I think, Krista, what else is out there that we just glossed over, and I didn't want to do that.
You know what? You answered so many questions. I did just drop in the chat, and it's already disappeared again. I'm going to drop the link to the Ask the Can-Do team in the chat again.
Good.
So if we did not get to your question tonight, please jump on there and check that out. I shared a lot of resources with everyone in the chat. There were a handful of questions about diet or gut microbiome, and we did have some amazing researchers. Dr. Alana Katz-Sands on in September. We had Dr. Walls on our webinar in August. So definitely take a look at some of our webinar archive as well that I think can help answer these questions and give some of you folks that want a deeper dive into some of these topics the option to do so. I will also give a shameless plug. Our first podcast of January is going to be about progressive MS research specifically. So I would invite you to join us and Dr. Bruce Bebo on that webinar, or I'm sorry, that podcast, to learn more about progressive updates specifically. And
our first webinar of 2024 will be about disease modifying therapies, not necessarily the research, we tried to cover that, but how people get started on a disease modifying therapy. If you have to switch therapies, what are some criteria that or questions that you can ask your provider if you're thinking, man, I don't know if this stuff is working. There's some questions that you can ask that we will go over. So there you go. Thanks for that, Krista.
That is the perfect plug, Kathy, because I feel like some of the questions that we didn't answer tonight were about specific symptoms for some of the DMTs. So what a great way to segue into our 2024 webinar series. With that, I do want to let you guys know a few housekeeping things. So this year, when we move into 2024, we're actually going to be rolling over our webinar registrations. So if you are registered for the current 2023 webinar series, you do not need to re-register. We have taken care of that for you. If for some reason you changed your mind and you do not want to be registered for the webinar series moving into the new year, please feel free to email us at info@cando-ms.org, and we'd be happy to remove you from that. But we hope that you decide to stay with us for many years to come.